To understand the origins of the spinal and cranial nerves, we will draw the developing embryo. First, draw a coronal section through the dorsal half of the embryo. Label the outer layer as ectoderm. It derives the epidermis and neuroectoderm. Then draw the endoderm at the bottom of the diagram. It comprises most of the ventral half of the embryo, and it forms the gut and respiratory contents of the developing embryo. Next, draw the notochord and indicate that it is mesoderm derived. We have thus established the three main layers of the trilaminar developing embryo: the ectoderm, mesoderm, and endoderm. The embryo forms around the notochord, and the notochord eventually degenerates into the jelly-like substance of the intervertebral discs, called nucleus pulposus. If the notochord persists in its primitive state, it is considered chordoma, notochord tumor. The notochord induces the overlying ectoderm to develop into the neural plate. Its neural folds then invaginate to become the oval-shaped neural tube, which has a long, narrow cerebrospinal fluid space in its center, and as mentioned, is ectoderm derived. We will draw the details of the neural tube later. Dorsolateral to the neural tube, draw the neural crests, which are neural tube derivatives that give rise to the peripheral nervous system. Next, draw somite tissue masses lateral to the notochord. They, like the notochord, are mesoderm derived. The neural crest cells and somite tissue masses develop early in embryogenesis, and the somite masses derive sclerotomal, myotomal, and dermatomal cells. We will include the somites along with their derivatives, albeit an anachronism, for simplicity. Choose a somite mass and draw 3 arrows from it as follows. Direct one to the notochord and label it sclerotome. The sclerotome differentiates into bone. Indicate that the sclerotome around the notochord becomes vertebral bone. Draw another arrow dorsally to the surface of the embryo and label it dermatome, which derives the dermis, the skin layer underlying the epidermis. Direct the last arrow laterally to form myotomal masses, which derive skeletal muscle. An artery and nerve pair corresponds to each somite segment, and wherever the products of that somite travel, its nerves and arteries follow. This is the anatomical basis of how we systematically assess dermatomal and myotomal levels during the neurologic exam. From the peripheral distribution of the deficit, we localize the rostrocaudal level of the lesion within or peripheral to the central neural axis. With that embryogenesis as a background, let's take a closer look at the origin of the cranial and spinal nerves. Inside the neural tube, label the ventral basal plate and the dorsal alar plate. Along the horizontal meridian, draw the sulcus limitans, a small sulcus which cuts into the lateral walls of the fluid space. The sulci separate the basal and alar plates. Somite nerve pairs emanate from these neural plates as nerve roots. According to the law of Bell and Magendie, ventral roots are motor and dorsal roots are sensory. This functional division applies to the neural tube as well. The basal plates, which produce the ventral roots, house motor cell columns, and the alar plates, which receive sensory roots, house sensory cell columns. The ventral motor dorsal sensory division that exists early in embryogenesis in the neural tube persists throughout development, and it is substantially preserved in the adult spinal cord and brain stem. It provides the basis for the organization of the spinal and cranial nerves. This concludes our diagram of the spinal and cranial nerve origins.